Universitas Bengkulu, universitas bagi berbagai suku, adat, dan budaya untuk membentuk karakter dan profesional di bidangnya. Selama berdiri, Universitas Bengkulu telah meluluskan wisudawan dan wisudawati terbaik di bidangnya masing-masing. Tentu Anda membutuhkan bakat, dedikasi, dan kerja keras. Jadi, selamat bergabung di Universitas Bengkulu. Sebagai mahasiswa, di sini Anda akan menemukan berbagai program studi yang akan membawa Anda bersaing di dunia industri nasional dan internasional. Sesuai visi kami untuk menjadikan Universitas Bengkulu berkelas dunia pada tahun 2025.
Mengembangkan pendidikan dan penelitian merupakan misi utama kami. Kami tidak henti-hentinya mendorong seluruh sivitas akademika Universitas Bengkulu untuk mempublikasikan karya ilmiahnya di jurnal internasional bereputasi dan dalam bentuk hak atas kekayaan intelektual yang nantinya bisa diaplikasikan dalam pengabdian kepada masyarakat di skala lokal ataupun internasional.
Dengan tenaga pengajar berkualitas, terdiri dari 866 dosen dan 54 diantaranya adalah guru besar. Ini adalah bukti bahwa Universitas Bengkulu siap membentuk tenaga profesional yang berkarakter, berwawasan kemajuan, dan berdaya saing. Untuk menjawab tantangan perkembangan zaman, kami terus membangun fasilitas-fasilitas penunjang bagi mahasiswa, bukan saja untuk membentuk karakter, tapi juga sebagai wadah penelitian yang nantinya bisa bermanfaat untuk masyarakat luas.
Universitas Bengkulu adalah salah satu universitas yang penuh dengan kebinekaan. Berbagai macam suku bangsa ada di Universitas Bengkulu. Ada dari Papua sampai ke Aceh. Semua berbaur menjadi satu di Universitas Bengkulu. Universitas Bengkulu telah mendidik mahasiswanya dengan baik, sehingga memiliki kompetensi yang hebat, memiliki daya saing yang tinggi, bahkan telah berpartisipasi banyak dalam event-event nasional, event regional dan internasional, dan sudah banyak sekali prestasi yang ditorehkan oleh mahasiswa Universitas Bengkulu.
Untuk menunjang akademik, baik di sisi pendidikan, penelitian, dan pengabdian kepada masyarakat, kami sudah melakukan beberapa kerjasama seperti dengan instansi pemerintahan provinsi, instansi pemerintah kabupaten dan kota, perguruan tinggi negeri dan swasta, baik itu dalam dan luar negeri, serta dunia industri dan dunia usaha.
Saya Muhammad Fajri Hidayat, selaku Ketua Laboratorium Kehutanan Universitas Bengkulu. Perlu kami informasikan bahwa Laboratorium Kehutanan Universitas Bengkulu itu sudah lama berdiri sejak tahun 1992, dan sampai saat ini sudah banyak melayani kegiatan akademik, penelitian, dan juga kegiatan pengabdian masyarakat yang dilakukan oleh sivitas akademika jurusan kehutanan dan juga Universitas Bengkulu pada umumnya. Oleh sebab itu, Laboratorium Kehutanan Universitas Bengkulu kami bertekad ke depannya untuk senantiasa berbenah diri, terutama dalam meningkatkan layanan prima laboratorium kepada mahasiswa, kepada dosen, kepada peneliti, dan juga kepada stakeholder lain yang berada di luar Universitas Bengkulu untuk melakukan kegiatan-kegiatan saintifik di Laboratorium Kehutanan. Oleh sebab itu, Laboratorium Kehutanan sudah menegaskan visinya di mana visi daripada laboratorium kehutanan adalah terwujudnya layanan prima laboratorium untuk menunjang kegiatan akademik dan kegiatan pembangunan sektor kehutanan pada umumnya. Sesuai dengan tuntutan untuk menuju laboratorium yang profesional dan handal, maka laboratorium kehutanan saat ini memiliki 4 divisi. Yang pertama adalah divisi manajemen hutan yang dikoordinasi oleh Bapak Dr. Gunggung Seno Aji. Kemudian ada divisi ekologi konservasi yang dikoordinasi oleh Bapak Profesor Dr. Agus Susatya. Kemudian ada divisi silvikultur yang dikoordinasi oleh Bapak Insinyur Putranto Budiono Agung Nugroho. Dan terakhir adalah divisi pengolahan hasil hutan yang dikoordinasi oleh Bapak Profesor Dr. Ridwan Yahya MSc. Untuk mendukung layanan dan servis yang akan diberikan laboratorium kepada pihak luar dan kepada mahasiswa, maka kami memiliki 2 laboran yang saat ini senantiasa kami persiapkan menjadi laboran yang handal dan memiliki juga 1 tenaga administrasi yang digunakan untuk melayani kebutuhan administrasi laboratorium, baik itu oleh mahasiswa, dosen, dan juga stakeholder lain yang di luar yang akan menggunakan jasa laboratorium. Untuk mengimbangi kemajuan teknologi, ilmu pengetahuan dan teknologi di bidang kehutanan, maka kami selalu senantiasa berbenah untuk senantiasa meningkatkan layanan-layanan laboratorium yang bisa kami lakukan, termasuk juga analisis dan layanan jasa lainnya. Pada saat ini di laboratorium kehutanan kita bisa melakukan seperti misalnya adalah jasa identifikasi jenis tumbuhan, pohon, dan juga tumbuhan bawah. Kemudian juga kami juga bisa melakukan estimasi dan pengamatan satwa liar. Dan saat ini di laboratorium kehutanan sudah juga bisa melakukan layanan yang terkait dengan analisis tanah, seperti misalnya sifat fisika tanah, sifat kimia tanah, dan juga kami juga sudah bisa melakukan analisis karbon, sifat fisika kayu, sifat kimia kayu, pembuatan pulp, dan juga saat ini kami juga sudah memiliki studio analisis spasial, yang dimana studio tersebut adalah untuk membidangi jasa-jasa yang bisa kami berikan terkait dengan sistem informasi geografi. Jadi di laboratorium kehutanan saat ini sudah bisa melakukan analisis spasial yang juga cukup banyak, seperti misalnya pembuatan peta dasar, pembuatan peta tematik, kemudian pemodelan yang banyak digunakan di bidang di sektor kehutanan. Seperti misalnya kami sudah bisa membuat peta-peta tematik terkait dengan lahan kritis, gitu, pemodelan banjir, pemodelan longsor, kemudian dan pemodelan-pemodelan lainnya yang terkait dengan analisis spasial. Jadi dengan jasa layanan yang bisa diberikan, kami berharap bahwa ada dukungan dari semua pihak agar supaya laboratorium kehutanan itu kedepannya akan makin berkembang menjadi laboratorium yang memiliki layanan prima, profesional, dan handal. Saya kira demikian. Terima kasih.
it will be a long Chris. Pak Agus, Professor Agus, he's the expert of Rafflesia. We would like to have a long collaboration in research in plant species. Dr. Wahyudi problems with the forest because some of the area has been mined for the coal, so needs
So the Botanic Garden is quite small, and it was established by someone called the Earl of Danby, and he provided a sum of money to protect an area of land by the river to grow medicinal plants, to teach Oxford University's medical students about the medicinal properties of plants. So it has a long 400-year history. And today, like other botanic gardens, we have a wide scope. So we grow plants for research, we grow plants for conservation and for teaching purposes. And also, importantly, our botanic garden is a place of recreation and important for people's mental health and well-being. So just like Bogor Botanic Garden, which I'll mention in a moment, it's a place that people come to for an oasis in the city. Green spaces are very important for people's mental health and well-being. And so sometimes I like to think that Oxford Botanic Garden, that was first established as a place of human healing, a medicinal garden, is in a way still a place of human healing, but more for the mind than necessarily medicine in a conventional sense. So I'm very fortunate to work in this beautiful place that has a very rich history. Next slide, please. And this year we celebrated our 400th anniversary. Oh, thank you. That's very useful. Great. This year we celebrated our 400th anniversary. This is our Chancellor of the university. This is my boss, Professor Simon Hiscock, and then here he is again, and me processing down the high street, celebrating our 400th anniversary this year. And the reason I share this is because on the next slide, please, and if you could kindly press play, this is a little, if you press play, it's a video. I think if you hover your mouse over the slide, a little play button comes up. Whilst that loads, what I was going to share with you is that whilst we were celebrating our 400th anniversary, Bogor Botanic Garden No, not this one. No, that's the end. No, don't worry. What I was going to share with you was a video from Joko showing the celebrations of the bicentenary in 2017 at Bogor Botanic Garden. Yeah, there you are. There you are. Thank you. I think if you hover your finger over the pad and then you can press play. There we go, there we go. So this has been sped up. So it's very, very fast. But we just wanted to give you a flavour of the celebrations that have happened at Bogor for the bicentenary. Very, very fast. Okay. Thank you. So I think it's very fitting that in Oxford we were celebrating an anniversary, and in Bogor they've been celebrating an anniversary. So it's wonderful that two historic botanic gardens come together in this broader collaboration. Next slide, please. Just a little bit about some of the research that we do at my institution. So these are some recent examples that we're very proud of that made the front cover of some journals. And our research is very broad. So today I'm going to talk specifically about Rafflesia, but we work on other things. So on the left you can see the underside of a giant water lily leaf. And this is from some research that we do into biomimetics. Does anyone know what biomimetics means? Exactly. So biomimetics is in technology where we copy design in nature to inform technology. So for example, we've moved on. If we go back one. Yeah. So on the left, this shows the underside of a water lily leaf, which we did some research to show that this can inspire the design of floating solar panels. Because as I say to my students, what better source of inspiration could there be for technology than a solar panel that has been tried and tested by evolution for hundreds of millions of years? It's very, very efficient. And then in the middle, you can see some examples of molecular ecology work that we do. And then on the right, some public engagement work, which I'll talk more about later. Next slide, please. So before I talk about Rafflesia, I wanted to talk more generally about the flora of Southeast Asia, because I have to say that your flora that you have in Southeast Asia is the envy of people around the world. I think you have the most magnificent flora of any region. And that's something that I've always thought. So when I was small, I used to have books on nature, plants and animals. And I remember looking at pictures of some of the plants and animals that live in Southeast Asia, and I was mesmerized by the wonderful and very rich nature that you are so lucky to have here. This is not, you're very, very lucky, because this amount of nature is not normal around the world. So you share your home with a wonderful part of the biosphere. Yeah, so I've been very lucky as a botanist to dedicate some of my research to these extraordinary plants that grow here. So I'll let you know when to go next. Thank you. So this is a Nepenthes pitcher plant, Nepenthes hemsliana, which grows in Borneo. And this is a species of Nepenthes. The pitchers do not trap insects like most Nepenthes. This one is a home for bats. And so the bats have evolved a beautiful symbiosis with the pitcher plant. And what happens is the bats roost inside the pitchers, and their droppings, the defecation, nourishes the plant, and the bats in return have a form of sort of sheltered accommodation, a safe place to sleep and to rear their young. So this is a beautiful mutualism that has evolved between a plant and an animal. And when I was a small boy and I looked in my picture books at the plants that grow here, this was not known. This is a much more recent discovery by scientists. So today we're still discovering more and more about the unique flora that grows in this part of the world and how it interacts with animals. Very, very interesting. Next slide, please. Whilst I'm talking about celebrated plants in Southeast Asia, of course you need no introduction to this one. So these are durians, of course, and I had the great privilege of working with Jocko in the last year on a paper that was a brief review of how celebrated the durian fruit is in Southeast Asia. So we looked a little bit at new species descriptions, and how much this fruit means to people in Southeast Asia. A very, very much celebrated fruit, as you know, called the king of fruits. And on the next slide, you can see that we had the great pleasure of sampling a durian on our travels here in Sumatra. Generally, people say that Western people like me do not like durian. However, I have grown over the years to very much appreciate durian. I must say that the first time I tried it, it was very strange to me, because it tasted like onion or garlic, maybe, with a strong smell. But over the years, I have tried durian many, many times, and now I feel that I truly appreciate the complexity and the very fine, sweet flavour of durian, and we very much enjoyed eating our durian this week. As well as some new things for me. So Chempedak. This is new to me. So I've travelled quite a lot in Southeast Asia, but I haven't come across this fruit before. And when I shared this on social media, lots of people commented that to them it looked like a strange exotic sandwich with lots of filling. But it had a very rich and unusual flavour. So as well as a wonderful flora that you have in Southeast Asia, you have a very interesting diversity of fruits that are very much celebrated. So it's very interesting for people like me as a botanist to visit your home and to see these wonderful and strange fruits that are very much celebrated here. But of course, the greatest enigma and the jewel in the crown of the flora of Southeast Asia, I would say, is Rafflesia, an enigmatic, strange, mysterious and beautiful plant, which is the focus of our research here in Sumatra this last 2 weeks. It will be the focus of our research together, we hope, in the future, and it's certainly the focus of today's talk. So a very, very special plant that we have been so privileged to see in this last 2 weeks. So I'm now going to talk about parasitic plants more broadly. What are parasitic plants? So parasitic plants have evolved 12 times independently. These are plants that extract nutrients from other plants. So when we think of a plant conventionally, we think of something that is photosynthetic. All around us through the windows you see green plants, autotrophic plants. They're busy photosynthesizing with chlorophyll in their green tissues, capturing, harnessing energy from sunlight to manufacture food. But there are some plants that do not manufacture their own food, they steal it from the roots or sometimes the stems of other plants. And these have evolved 12 different times. And here are examples from around the world of those 12 independent origins of parasitic plants. But they have something in common. So all of them have evolved a specialized structure called a haustorium. Is a haustorium something that you have come across before in your studies? Yes. So a haustorium is a fascinating structure. It's a living physiological bridge between one organism and another, a cellular bridge, if you will. So each of these plants have evolved a way to penetrate the vascular tissues, the water and sap conducting tissues of their host, to extract nutrients into the parasitic plant. So it steals its food from other plants rather than manufacturing its own through photosynthesis. Now there are parasitic plants that do photosynthesize, and these are called hemiparasites. Mistletoes are good examples if you're used to those. Please, back to the slide we were on. I will indicate when to move. Thank you. Hemiparasites are photosynthetic, but the examples that I'm showing here are hollow parasites. Hollow parasites do not have chlorophyll. They do not have leaves or roots in many cases. They are very, very much reduced. So as they have evolved and become very derived in their life history, they have lost chlorophyll. They have lost photosynthesis, they have lost leaves, and they have lost roots. And for this reason, sometimes people think they look superficially like fungi, but these are not fungi. Fungi belong to a very different kingdom altogether. These are plants that have lost the features that we think of as being very characteristic of plants. What is also interesting about parasitic plants is that they have challenged taxonomic botanists for centuries. So we're joined by some very fine taxonomic botanists today, and they will share this view that for a long time these plants have been very much confused because taxonomic botanists like to observe details. Botanists like details. So we like to look at the shape of leaves, the characteristics of roots, all sorts of these features. But as parasitic plants have evolved, they have lost the features shared by their photosynthetic ancestors. And this meant that taxonomic botanists were bereft of interesting characters with which to classify plants. And this meant that these plants have been very much confused for centuries. And it was only with the advent of DNA sequencing technology that scientists have been able to assign these plants to the families. And so this grid, which shows 12 different lineages of parasitic plants, if you were sitting in this lecture theatre 30 years ago, it would not be possible to share this because it was not known what the ancestry of these plants was. We needed DNA sequencing technology to tell us objectively how these plants evolved and what their ancestry was. And so we know, for example, that Rafflesia, the largest flower in the world, the focus of this talk, the ancestors of these plants are plants called euphorbias, the Euphorbiaceae family. And what's really fascinating is that they have very tiny flowers. So actually Rafflesia has evolved to produce these enormous flowers from ancestors with very tiny flowers. And taxonomic botanists would never have known this if it wasn't for DNA sequencing technology. Next slide, please. There are other examples of parasitic plants, which I will touch upon briefly, and they are fungal parasites. They're also known as mycoheterotrophs. Have you come across these plants before? Are these familiar? I think possibly not. No, possibly not. So the previous slide I showed you examined plants that are parasitic on other plants. These are parasitic on fungi. Now, most plants have fungal associations, associations that we can't see. There is a symbiotic relationship that occurs underground between plants and fungi that visually we're unaware of. But sometimes there is an asymmetry in that relationship. So, for example, there are some fungi that are parasitic on plants. These are called biotrophic fungi. But there are also some plants that are parasitic on fungi, and these are the examples here. And just like the previous examples of parasitic plants, you can see that they lack chlorophyll, they have no green pigments, they lack leaves, they lack photosynthesis, and actually they lack functional roots. So in many respects, they're very similar to the plant parasites. It's just that they're extracting nutrients from a different organism. In particular, the photos I've shared here are from around the world. The one on the left is an orchid called Rhizanthella from Australia, and it only flowers underground. So scientists had not seen it for a long period of time. You have to dig it up to see the flowers. The next curious plant is one called Oxygyna, which grows in Japan and West Central Africa, so it has a disjunct distribution. And then the three on the right belong to the Thismiaceae family. And all of these plants are fungal parasites, so they are obligately dependent on a fungus for their nutrition. Next slide, please. Here are some more examples. So on the left are some examples of Oxygyna, fungal parasites that I mentioned from West Central Africa and Japan. And then on the right, some Thismiaceae from Malaysia. I think they have an extraordinary beauty. They're very curious. They're sometimes called fairy lanterns, is a common name for these plants. And they're very enigmatic. And I imagine that you have Thismia here in Sumatra. We saw some Bomaniaceae on our expedition this week, which is a distant relative of these plants. So a very mysterious group of plants. And I would encourage you, if you consider pursuing a career in botany, to dedicate time to some of these lesser known plants. Not very much attention has been given to them, and I'm sure there are lots of mysteries awaiting discovery. Next slide, please. This is one that I had the honor of describing with my colleagues on the screen. So this chap here on the bottom is a man called Domi Nikong, who is a wonderful rainforest explorer in Terengganu in peninsular Malaysia. And he made a discovery, this orange flower at the front, very, very small. My slide makes it look large, but it's smaller than my little finger. He discovered this plant growing in the rainforests, and he brought it to the attention of Siti Munira, a botanist in peninsular Malaysia, also on the screen. And then she brought it to my attention. And together, when we examined the features of this curious plant, it became very clear to us that this was a new species of Thismia, one that had not been seen before. And so we described this as new. What's very interesting about this plant is that since we described it, it has not been seen again. So it is critically endangered. It may already be extinct. These plants are very ephemeral. They're very unpredictable. It's very difficult to know exactly when they're going to flower. Rather like Rafflesia, in a sense, only with these, they're so small. With Rafflesia, you can hardly miss it. It's so large. But with Thismia, it's very difficult to see them. And sometimes they never even emerge above the leaf litter, so we'd never know they're there. So these are very, very difficult plants to work with. In the background are two other species of Thismia that occur in Malaysia. I think these are very beautiful. They're very strange, but very beautiful and very poorly understood plants. Next slide, please. Some of my work has focused on a particular family called the Orobanchaceae. We have a rich assemblage of Orobanchaceae in Europe, where I come from. Next slide, please. And just to note briefly, you do not need to see all of the detail on this slide, but the interaction between a parasite and its host is highly complex. So there are chemical signals released by the roots of a host plant that elicit germination, and there are further signals that elicit haustorial formation. So the haustorium, remember, is the physiological bridge between the parasite and its host. And there are chemical stimulants that trigger these different stages in the development of the parasite. And actually, we understand very little of those for most species of parasitic plants. And for Rafflesia, the focus of today's lecture, we understand almost nothing at all, because when scientists have tried to grow Rafflesia in vitro, it has proved very, very challenging. So the largest flower in the world is also the greatest enigma. So these plants are fascinating, but very difficult to work with. Next slide, please. And what happens at a cellular and at a molecular level has implications for what happens at a population and an evolutionary level. So this is a complicated slide, but I'll summarize briefly. Looking at Orobanchaceae, another parasitic plant, much smaller than Rafflesia, some research we did, we took some seeds from Orobanchaceae growing on one host, called a clover, and then we took seeds of Orobanchaceae growing on another host, called a wild carrot, a sea carrot. And we grew these seeds on the host on which we collected them, and also the other host. So that's something called a reciprocal cross, if you follow me. So two parasites, two hosts, and we grew each one in a reciprocal formation to understand how well these parasites could grow on alternative hosts. And the complicated pictures on the right show at a cellular level that parasites perform better on their local host, on the host on which you collect it, than they do on an allopatric host, on a host that you've collected from somewhere else. In other words, these parasites are locally adapted to a specific host. Why is this important, and what does it mean? What it means is that if parasites are locally adapted to a host, and if that host has a different ecology, it means that these parasites are becoming ecologically separated. This is a process called allopatric isolation. Is allopatry something you've come across before? Perhaps. Allopatric speciation is the divergence geographically of populations. They become genetically divergent over time. And so what we then did, after growing these on different hosts, is we looked at the population genetics structure of this. So something rather complicated called sequence-characterized amplified regions of certain bits of very variable DNA. We looked at those, and what we found was that the populations that are locally adapted to different hosts are also genetically different. And so even though these parasitic plants look the same, they are behaving differently. They are physiologically adapted to different hosts, and they are genetically divergent. And so if we think of Rafflesia for a moment, Rafflesia, as you, I'm sure, know, grows on a tropical vine called Tetrastigma. We know very little about Tetrastigma. It's a taxonomically very challenging group in itself. It's very difficult for us to identify Tetrastigma in the field. The species are very similar. They're not well characterized. It's very difficult to see the flowers because they occur in the canopy. So we understand rather little about the hosts of Rafflesia. Actually, we were interested to observe that in Bogor Botanic Garden, which I'll talk about our observations more, but we saw Rafflesia growing on different species of Tetrastigma. So it may be that Rafflesia can grow on lots of different species of Tetrastigma, but we know very little about this. And unlike Orobanchaceae, which I can grow in a petri dish relatively easily in a lab, Obviously, that is not the case for Rafflesia. It's not something that you can quickly grow in a petri dish for obvious reasons. And so our understanding is much more limited. So there is a lot more to discover still for Rafflesia. Next slide, please. I'd like to introduce you to another weird parasitic plant that you may not have encountered before. It occurs in Africa. So with my collaborators, Duncan Cameron and Owen Grace, we have a PhD student, Seb, who is looking at the evolutionary biology of this plant. Isn't this the weirdest plant you've ever seen? It's called Hydnora, and it grows in the deserts of Africa. And this is also poorly understood. So it grows about this big, so not enormous like Rafflesia, but like Rafflesia, you can see no chlorophyll, again a parasitic plant. And this one grows on the roots of desert shrubs called euphorbias. And on the next slide, my very capable student, Seb, has been busy examining lots and lots of different specimens and has just produced a monograph for the genus. And he discovered that looking at something called osmophore geometry, it was an underestimated important trait in this genus. And so we described 2 new species to add to the genus Hydnora. So these parasitic plants are often very poorly known to science. Hydnora, in fact, is among the most basal of parasitic angiosperms. I don't know how much information you've already covered in your syllabus about the angiosperm phylogeny. Professor Argus, is this something that the angiosperm phylogeny, is this something that maybe has been covered?
In the first year in biology.
Okay, great. Yes. So, right, so the angiosperm phylogeny is the family tree of all of the flowering plants. So what is related to what? And if you look at the very bottom of that, you will see the first plants to have diverged from the flowering plant family tree. And among them are plants called the Aristolochiaceae, and the relatives of which are Hydnora. And so we're very interested to examine this because these are some of the most evolutionarily primitive of parasitic flowering plants. And so maybe they hold clues for us about the evolution of parasitic plants more broadly. And so that's why we're interested in looking at this specific parasitic plants. And for the research scientists among us, specifically what we really hope to do is to produce a well-resolved phylogeny for Hydnora, and then to examine how the different nodes of the phylogeny grow on their different hosts at a physiological level. To use a combined approach to see how these plants are evolving. Next slide, please. This is just a quick reference to a relative of Hydnora. Doesn't it look like a fungus? But it's not. It's a plant. This is Prosopanchi, which that grow up to your knee, very large, very colourful. So parasitic plants are not just in rainforests, they're also in deserts. Next slide. And working with my collaborators, we are interested to know how this might be farmed and used in the future as a possible crop. So in China, this parasitic plant for thousands of years, it's called Rokong, and it has been harvested and used for traditional herbal medicine in China. And actually, the Chinese are now growing this parasitic plant at scale on shelter forests. Now, shelter forests are long lines of desert-tolerant trees that are planted to halt land degradation. So desertification and land degradation is a big problem in the deserts of the world. It's a major threat to ecosystems. And these long lines of trees are planted, they act like a fire break, so they stop the spread of deserts. And you can actually grow this parasitic plant on the roots of these shelter forests and then harvest it as a crop. So at the same time, as people are growing shelter forests in the interests of protecting the land from climate change, there is also the potential to grow a new crop that is useful in desertic areas where it's difficult to irrigate or to use fertilizers. So parasitic plants are also useful as well. Next slide, please. This just shows you the underground stem. Can you see the people in the background to give an idea of scale? So you can see just how enormous this plant is. It has a very, very large underground stem. Next slide, please. Next slide. Thank you. So now I will talk a little bit about the evolutionary biology of our big enigma. And this is a photo, by the way, that we took here in the field. Thank you. So on to Rafflesia and similar plants. So Rafflesia is a parasitic plant, and I've already introduced those to you, but it's more than just a parasite. It's what we call an endoparasite. So Rafflesia not only steals its food from the roots of other plants, it actually lives inside another plant, which is an extraordinary life history and one that is very rare in the plant kingdom. So endoparasitism in plants has only evolved 4 times. And on the angiosperm phylogeny, which I just referred to, I have mapped for you 4 different independent origins of endoparasitism. So there are 4 different lineages. I mentioned a few moments ago that botanists were once very confused about the evolutionary origins of parasitic plants because they look so different to other plants. But actually, when scientists looked at the DNA, they found that there are 4 different independent origins. So these 4 different types of plant that are on the screen are not related, but 4 times they evolved the same life history to grow within the tissues of a host plant. So here we have my pointer. Rafflesia, which you know, but we also have Mitrostemon, which is about the size of my finger. It's actually quite small. This one is called Cytinus, which grows in Mediterranean districts. And this one is Pilostyles, which belongs to the Apodanthaceae. And they all grow in different parts of the world, and they evolved independently. And now I'll show you a little bit more about the biology of these. Next slide, please. Rhizanthes. So Rhizanthes, same family as Rafflesiaceae. So Rhizanthes, Sapria, Rafflesia. So I've just put one representative from each of the four lineages here. And so working with my colleagues here, we were interested in this particular life history that is so uncommon in plants. So Rafflesia is extraordinary to look at. We all know that. This is much celebrated, largest flower in the world. But it's extraordinary for more reasons than just its size. It's also extraordinary because of its biology, because of its life history. And I know that this is something that your Vice Dean is very interested in, the life history of this parasitic plant. Particularly because it shows a level of cross-kingdom convergence. So remember, convergent evolution is when the same pattern has evolved more than once, usually in response to common selection pressures. So a typical example would be in America, you have a cactus. In Africa, you have something that looks like a cactus called a euphorbia, but it's not. They evolved independently in response to drought stress. Similarly, parasitic plants like Rafflesia show an extraordinary cross-kingdom convergence with fungi. So I'll show you how. So in the case of Rafflesia, it produces many, many tiny seeds that germinate in response to a chemical exudate produced by the roots of the host. But we know very little about that process, as I've said. Similarly, this fungus at the bottom, called Ustilago maedis, which is a a pathogenic fungus, produces many tiny spores that also respond to chemical stimulants produced by the host. Then the seed of Rafflesia somehow attaches to the tissues of its host. We don't know about this process. This has not been seen. It's very much a mystery, which is fascinating. And then in a fungus, it produces something called an appressorium, but it's in some ways similar. And then it goes undercover. So Rafflesia penetrates the vascular tissues of its host. And here you can see these cells. For most of Rafflesia's life, that is all that this plant comprises. It's extraordinary that the largest flower in the world spends most of its life as a series of... say again, sorry, inside the... This is in the cortex, yes, yeah. So it spends most of its life as a series of what we call uniseriate filaments. So effectively, undifferentiated chains of cells that live inside the tissues of its host. Similarly, these fungi produce threads that penetrate the tissues of their hosts. There are some differences because this one grows intercellularly and this one grows intracellularly. So if you understand me, the Rafflesia grows between the cells and the fungus grows into the cells. But to all intents and purposes, they share a commonality in that they go undercover and they exist inside their hosts. And actually, the level of convergence goes much further than beyond what you can see down a microscope. Because of this intimate association between the host and the parasite, there is a high level of communication at a molecular level between the two organisms. So what I mean by this is that over the course of evolutionary time, Rafflesia has acquired genetic material from its host. This is a phenomenon called HGT, horizontal gene transfer, and I can see some nods, so perhaps this is something you've come across before in your syllabus. So we tend to think of genetic material as being inherited, passed down vertically down the germline, but just once in a while, genetic material can be transferred laterally by something called horizontal gene transfer, and that's really, really fascinating. And Charles Davis on the top here and his team have looked in detail at this, and they found an elevated amount of crosstalk between host and parasite. And within the genome of the parasitic plants like Rafflesia and its relatives, they contain what have been called DNA fossils. So DNA from former host associations. So Rafflesia today only grows on tetrastigma in the Vitaceae family, the grapevine family. But historically, long before we're sitting here now, so millions of years ago, Rafflesia and its relatives will have grown on different hosts beside tetrastigma. For example, in the Cucurbitaceae, the cucumber family. And we know this because they have remnants in their genome still today called DNA fossils. And it's not just historic crosstalk between host and parasite. There is probably still a high level of molecular communication between host and parasite. So if we look in better known examples such as fungi, we see fascinating examples of molecular camouflage. So as the parasite goes into the tissues of its host, it plays all sorts of molecular tricks on its host to remain hidden. DNA silencing and small RNAs, non-coding bits of RNA that can influence what's going on at a molecular level to remain hidden from the defense mechanisms of the host, which is really, really very fascinating. And to my mind, I think it's likely that there's a lot of this going on in Rafflesiaceae because it's not within the interests, if you like, of a tetrastigma vine to have a big parasite like Rafflesia growing inside it. And so, to my mind, that suggests that there is a lot going on at a molecular level that is obscuring the identity of Rafflesia inside the host. The host does not know, in inverted commas, that the parasite is in there. And I think that is probably due to some of this crosstalk between host and parasite, keeping the parasite quiet at a molecular level so that the host cannot resist it and kill the parasite inside. But we don't know this yet. This remains very poorly understood. Another mystery of Rafflesia. Finally, after an extended period of pro-embryonic growth, and we believe from our conversations with colleagues here that from seed to flowering, Rafflesia probably takes 10 years, is our understanding from Sophie Musa Duarte in Bogor. So a long period of time spent within the tissues of its host as a series of cellular filaments. It then does something remarkable, which you all know. It produces these enormous flowers. So Rafflesia bypasses the vegetative growth phase that most plants have. So most plants, a seed germinates, it grows some roots, it makes a stem, it grows some leaves. Not Rafflesia. It has bypassed this vegetative part and it exists as a prolonged pro-embryonic state, so as a very, very reduced endophyte, and then it suddenly goes to reproduction. So its life cycle is very unlike other plants. And then just like the fungus, in the case of Rafflesia, it produces many, many thousands of tiny seeds, and of course the fungus produces many spores. And so if we simplify the life cycle of Rafflesia and a biotrophic fungus, we see there are lots of similarities. And so this is a nice example of a cross-kingdom convergence between plants and fungi, because as you know very well, fungi belong to a different kingdom to plants. They're not related. Next slide, please. And because I know some of you are interested in the life history and the biology of Rafflesia, I included this slide to show, if you look at a cellular level, as we have, of, and here you have your rhizanthes, and the other three lineages of endoparasites, there are some analogies. So each of these endoparasitic lineages exists as a reduced mass of cells within the host plants. They are a little different because in Rafflesiaceae it's very much reduced. In these other examples, and I know you cannot see from where you're sitting in detail, but hopefully you can see they produce these strange spikes that I've delineated in red. These have been described as sinkers. And what this means is that the endophyte, the strings of cells that exist within the tissues of the host, send down these sinkers that penetrate the vascular tissues of the host and extracts their nutrients and water from the vascular system of the host. And these structures have evolved three times independently. So very, very similar biology. What's also very interesting, just as a quick aside, is that in each of these lineages, actually, there is a great distortion of the host's anatomy when the parasite is living within the tissues of the host. So, for example, what you see is that if you have a tetrastigma vine with Rafflesia growing inside it, it causes a distortion of the xylem, the vascular tissues in the host. And we've called this something called hydraulic safety. So there seems to be a trade-off between, on the one hand, if you're a parasite, you don't want to reduce or compromise the biology of your host because that would not be in the interests of the parasite. And so distorting the anatomy too much is not a good thing. But on the other hand, if you're going to produce something as enormous as a flower like this, you probably need quite a lot of vascular tissue around you, so something called hydraulic safety. So again, it's another example of a trick this parasite plays on its host. It alters the micromorphology, the anatomy of the host in which it lives. Next slide, please. Now I'm going to talk about something slightly different. I'm going to talk about pollination biology. So this is a video. I don't know if it will work. It might do. Maybe it's too much. Oh, here we go. So this is a video I took in the Botanic garden where I work of a different plant, an aeroid. Oh, it's frozen. But what this video would show you, this very ugly, ugly plant, attracts flies. So like Rafflesia, this is called Helicodicerus muscivorus. And like Rafflesia, it produces a terrible smell. And like Rafflesia, it mimics rotting meat to attract pollinating flies. And on the next slide, my colleagues and I were very interested to understand why different species of aeroid, plants in the Aeraceae, have different smells, because we grow lots of different aeroids at the Botanic Garden and they have different smells, and maybe you have the same for your Amorphophallus species. I have not smelt enough to know. They smell different, and so we were very interested in this, and we wanted to know how is what we can smell different at a chemical level. And so we looked at these plants which appear to mimic carrion, dead meat, and some other plants which appear to mimic animal droppings or dung. And we wanted to see, are they chemically different? And on the next slide, you'll see some preliminary data. So we use a technology whereby we extract the chemicals from the smell, the volatiles, into a metal tube, and then they become frozen there for a period of several months. And then in a lab, we elute those chemicals with chemicals. It's called desorption. And then using mass spec, we then identify what are the different chemicals in those smells. And to our interest, what we found is that the ones in red, which mimic the smell of dead meat, have different chemical signatures to the species that mimic dung in black. So there are different chemicals involved in the mimicry of dead animals to the mimicry of dung. But not only that, these chemical signatures are species specific. So for example, this one here produces a very different chemical signature to the ones around it. So to our noses, we smell the difference, and of course that is happening at a chemical level. And on the next slide, just to make a note that we are, so Rafflesia, as you know, is pollinated by flies. So like the aeroids that I just shared, it mimics the smell of dead meat to attract flies for its pollination. This is a phenomenon in evolutionary terms called sapromyophily. And again, it's evolved multiple times in plants. A horrible smell to attract flies. And on the next slide, just to note that Adrian, Pat and I have been extending this work to Rafflesia. So Adrian has been busy traveling up and down the Philippines, sampling the smell of different Rafflesia species to see, are there differences in the chemical signature between these plants? And I'm almost certain there will be, because when we have smelt these different flowers, they smell very, very different. And actually, when we've visited the flowers in Indonesia, we noticed the difference. So to me, Rafflesia patma smells very different to Arnoldii, and that smells different to Bankuluensis. They have different smells and different strength, because patma was terrible smell. Arnoldii not quite so strong to our noses. Bankuluensis is pretty bad. So we have become experts in the terrible smell of Rafflesia in our fieldwork. And perhaps we will have the opportunity to extend this work to the species in Indonesia, which I think would be very interesting. And then we can look at the chemical signature of these different species. And just perhaps the different smells are attracting different pollinating insects. We don't know. Next slide, please. And again. Next slide. So on to taxonomy. So Rafflesia is very difficult to study taxonomically. Why? Well, one reason is that, forgive me, but it makes terrible herbarium specimens. I'm sure you have nice ones, but by and large, they don't make good herbarium specimens. It's not like leaves or flowers that press nicely. This is a big monster of a plant. It's a big fleshy thing, and it doesn't preserve well. So your choices are either to squash it or to stick it in a vial of ethanol. But either way, it doesn't preserve well. And so when we want to return to a type specimen years later after someone has described something, we cannot see it in the same way that someone's seen it, the living material. And that makes our lives very difficult as botanists. And that is probably part of the reason there has been a lot of confusion and discord among botanists as to what is what. Next slide, please. And so this great work by Jamili Nays, which you possibly know, I know some of you will, Rafflesia of the World, since this volume was published, we now have a further 13 taxa described from the Philippines, for example. So when this book was published some 20 or so years ago, there were just 2 species from the Philippines recognized. And we now know that the center of diversity of Rafflesia is in the Philippines. So I would say Bengkulu, it's the capital of Rafflesia in some respects. You have many fine species and the largest in the world. But in terms of the number of species, actually the Philippines has a very high number. Next slide. So here we've just summarized for you, and we just produced this slide together yesterday. And it's a crude estimate. It's not intended to be particularly detailed, and it's not weighted by geographic area. So in some respects, it's not accurate. But we just wanted to show you the current understanding of Rafflesia diversity across different regions, and this was extracted from multiple sources. Next slide. So I had the very great pleasure a few months ago in joining Professor Pat and Adrian Tobias across Luzon Island in the Philippines to examine some of the wonderful Rafflesia species that occur there. And so you can see there is a great diversity. And this has only been known about for the last few years. And in a few slides' time, we'll talk about why we think that means that Rafflesia is understood in a different way to here. And again, next slide. This is Rafflesia panchoensis. Maybe you can see from the photograph just how small it is. It's tiny compared with your enormous Rafflesia. This one is about the size of a human hand, very, very small. But perfect, as you can see from the photograph. A perfect, a pocket-sized Rafflesia, if you will. Next slide. But again, Rafflesia is very much confused in this region. So here is an example of 2 species that we have spent some time examining. So Pat first discovered this species in 2009. 2009 in northern Luzon in Kalinga. And since then there's been some confusion between this species and Leonardi, which is a very different species. As you can see, it has no processes, and if you dissect these plants, they look very, very different at a micromorphological level. And so what we've learnt here in this process is that it's impossible to understand Rafflesia taxonomy from photographs alone. And I know this firsthand because when I saw photographs of some of the species in Indonesia, to me they looked quite similar. And when we've examined them in the field in the last 2 weeks, it's very clear to us that they are very different. So a good rule of thumb is do not try and identify Rafflesia from photographs alone. It's very important to observe them in the field and look at their micromorphological features and the stable features. Next slide. So here is the magnificent Rafflesia banoana in the Philippines. And on the next slide, I have a video. I hope it will work, because I wanted to share with you the terrain that we crossed in the Philippines. Very, very difficult. So here, your Rafflesia are relatively accessible. So a short trek into the rainforest and you can find Rafflesia. Here, you have to trek sometimes for days through very difficult terrain and with lots of help from local people to get through the vegetation. So very, very different experience. Next. This is next slide. I've probably covered this. This beautiful work was done by Adrian, sitting over there, who has spent a lot of time examining and dissecting and observing the plants that Pat first discovered some years ago, and demonstrating that it's different. So this is a paper that we've recently submitted. And on the next slide, you'll see that Adrian and Pat have encountered on their fieldwork yet another new taxon. So this, and maybe after the presentation, if you'd like to know about this, do speak to Adrian and Pat about their observations of this plant, because it is indeed quite different. So I think this really shows you how little we understand about Rafflesia in some parts of the world. And again, I just wanted to share briefly something about me. So as well as a scientist, I'm an artist, and so when I visit different places in the world, I like to capture some of my experiences. And so these are drawings that I did of people that I met and encountered in the Philippines. And in the next few months, I will spend time doing the same for the people that I've met in Indonesia. So, so it will keep me busy. And again, next slide. Propagation. So I just briefly want to talk to you about something we've learnt from our time in Bogor. So I'll go through these slides quite quickly. Next. Some parasitic plants can be propagated relatively easily. So this is Orobanki, the plant which I told you I've worked on before. And as you can see, you can grow it in a petri dish and you can observe the whole life cycle within about 8 months. Within under a year, you can observe it from seed through to seed set. If only we could grow Rafflesia in a giant petri dish and understand it. But of course we can't. And so it makes it so much harder. But on the next slide, the world leaders in growing Rafflesia propagation, are in Indonesia. I say world leaders, dare I say almost the only people, I think, to grow Rafflesia. There maybe are some isolated examples elsewhere, but the only people to consistently grow Rafflesia time and time again are in Bogor Botanic Garden. And that was one of the primary reasons for our visit from the UK and the Philippines, was to learn from the experts in Indonesia how to propagate this plant, because there are other parts in Southeast Asia where we very much need to grow these plants for conservation. And so I had the great pleasure of working with Joko and his colleagues at Bogor Botanic Garden to publish a paper on how to grow the ungrowable, how to grow plants like Rafflesia, and to learn from that expertise. And learn we must, because Rafflesia needs protecting. And I think on the next slide... Oh. Oh, sorry, Yes, sorry, I forgot. Here is a brief video of Sophie Mursadawati's triumph. Is it going to work? Isn't this spectacular? Here you can see the unfolding of a miracle. This is Rafflesia patma growing at Bogor Botanic Garden, and this is Joko's friend who did the time lapse and the filming. Isn't it extraordinary? Wonderful. Yes, and on the next slide, this is a photograph that Adrian took of Rafflesia camarinensis in Luzon in the Philippines. And you can see here extensive habitat degradation. So this species is very much threatened by habitat degradation. And then on the next slide is another one, Rafflesia ballatei, which has an area of occupancy of under 100 meters squared, I believe. Of natural habitat left. So some of these species are under severe threat of extinction. And that's really worrying, because there are some plants that you can collect seed and you can grow it somewhere else. You can put it in a seed bank. You can't do that with Rafflesia. It's much more difficult. And so once it's gone in its natural habitat, it's gone forever. And so it's so important that we learn to conserve these plants. And so one of the primary reasons of our visit is to learn from the expertise at Bogor Botanic Garden how we might propagate and conserve Rafflesia in other places. Because it would be wonderful if, for example, some of these rare taxa in the Philippines could be grown in land grants, reserves, botanic gardens in the Philippines, or indeed in other places, to establish conservation collections of these plants, because time is running out for some species. Next slide. Indeed, we've seen a similar situation in Rafflesia in Indonesia. So this is a population we were kindly shown of Rafflesia bengkuluensis here in Indonesia, which grows on private land. And you can see already the encroachment of habitat destruction. And just a few meters into that habitat is Rafflesia bengkuluensis. And so you can see how important it is for us not only to protect these plants, but to understand more about them so that we can inform protection efforts, which I'll talk more about in a moment. So when Joko and I wrote this paper, we outlined some potential recommendations, but actually this visit we've expanded on these, as I will talk about in a moment. But just briefly, we recommend to extend successful propagation beyond Bogor Botanic Garden, the only place where this is done, to other places in Southeast Asia to protect Rafflesia. To understand how we can grow the plant from seed. So Sophie managed to grow Rafflesia arnoldii at Bogor Botanic Garden from seed, but it took 10 years, it's difficult. And then other in vitro protocols might be useful as well. Next slide. Now I'm going to share some very quickly some imagery from our wonderful expedition. This is a bud of Rafflesia patma at Bogor Botanic Garden. so an emblem of success. Here is Mr. Nagatari and Sophie Musadawati, and we are so very grateful for them sharing so generously their expertise on the grafting of Rafflesia. And what we now hope to do is to take those learnings back to the Philippines, because Professor Pat is the director of a land grant in the Philippines, a big reserve where tetrastigma grows wild, and we hope to be able to grow Rafflesia there. We've had a kind reception at Bogor Botanic Garden Herbarium, which was wonderful, and here at your own university, where we've been made to feel so welcome. Yes. We're so happy to be here. Next slide. And then locally at the ministry in Bengkulu. And we saw Rafflesia patma in the wild. Here is Dr. Joko with Rafflesia patma in Pangandaran. Rafflesia, and then Rafflesia Kumumu, which Deki Andriki waves his hand at the back, was so kind to share with us. Here he is. Deki is a hero. I'm sure many of you know Deki already, but he cares very deeply about these plants and wants to share them with people and share the wonder of Rafflesia. And he is the one that made contact with us and brought us together. So a big pat on the back for our hero, Deki. Terima kasih. Next slide. The wonderful Rafflesia arnoldi. And because people care so deeply about Rafflesia here, you can see that when there was a misadventure, as we say in English, it has been stitched together by the local people, because everyone cares so deeply here about Rafflesia, we know. And again, next slide. The beautiful and enigmatic Rafflesia benculuensis. It's like seeing a ghost. And I'd like to spend a moment congratulating these people as well. So during our trip to Indonesia, we've been fortunate to meet many people who are very passionate about Rafflesia, and it's very clear to us just how much people care. So this is Bang Zul, the former village leader of which village? In Kawod, somewhere in Kawod, who kindly took us to see Rafflesia benculuensis. And then this is Yogi and his group, his team, who also in Kawod locally make very great efforts to conserve Rafflesia. And this is deeply important, because unless we foster a deep care and attention for Rafflesia, we will not be successful in preserving it in the face of major changes to habitats. It's so very important. And we were so heartened to meet these people who care deeply about this very special plant. Next slide. So where next? What are our next steps? Well, that's partly why we're here, to talk to you about this, but our proposed next steps are: we would like to produce a revised phylogenetic framework. So at the moment, there are lots of different botanists around Southeast Asia working in isolation on Rafflesia, because they are the local experts, and that makes sense. But what also is helpful is to bring together effort to align and coordinate, so that we have a consistent approach and understanding to the taxonomic diversity of Rafflesia. Because, as I put in one of the last slides, chaos is the law of nature, order is the dream of man. We want to make sense of the living world around us. That's what taxonomic botanists do. We want to bring order to the chaos around us. But it's important that we do, because unless we understand what is there, we cannot adequately conserve it. So phylogeny and taxonomy is important for taxonomy, for conservation. It's not just important in its own right. Next, as I've already said, we would like to extend learnings across regions. We would like to take some of the information and the wisdom that we have received from Indonesia to deploy that locally in the Philippines. And we would like to establish a dedicated community of purpose, and that's what some of our conversations have clearly led to already, is that we're very keen and committed to working together to take some of this work forward. So that's the first thing, is to understand more, and then on the next slide, to understand and then to protect. So let's identify opportunities for sustainable tourism. So tourism is a double-edged sword. It carries risks because there are risks of trampling in natural habitats, but it also generates revenue to support local groups who, as I've said already, care deeply about Rafflesia and want to protect them. And it's a valuable source of income for that effort. And so if managed carefully, we suggest that this is a very useful strategy going forward. We want to strengthen and align and coordinate effort among local conservation groups. But actually, I have to say, I think this is something that's going on very well already. So when we've spent time with Deki, he spends a lot of time on his WhatsApp groups and his Facebook groups, talking to people up and down Bengkulu who know when a Rafflesia is about to flower, and everyone is talking to one another. And so there is very good coordination already. But that's not something that necessarily happens elsewhere in Southeast Asia. And then so important is to extend the protected areas. And I cannot emphasize this more. Some of the Rafflesia, the beautiful Rafflesia that we saw on this expedition, are in small unprotected areas of private land. And we would like to do everything we can to extend areas of protection, to give these plants the official status that they deserve, so that in many years to come, we're still having these wonderful discussions about these plants that still exist today, because we all care so deeply about them. So we do need to make effort to extend these protected areas. And so just some ideas that we've come up with together. Where first we need to identify where those areas are. You know where these areas are already, you know where the Rafflesia grow. And then we need to protect them. So it could be through the establishment of local conservation areas, critical habitat areas, and working with local government to make sure that these populations have the status that they deserve. And then efforts to seek funds. And this is what we as a collective can understand. What are the opportunities to seek funds to protect Rafflesia? Because we can't just conserve plants on fresh air. We need to bring in income to support this endeavour as well. Next slide, please. Something that is truly important, perhaps more than it may come across, is engaging people with Rafflesia. So these images here are something that we introduced at our Arboretum at Oxford Botanic Garden to show people how conservation action is important for plants, not just animals. So if you speak to someone in the UK, and I imagine it's similar here, and you say to someone, what does conservation mean? Tell me about conservation. They will tell you about rhinos, about whales, about elephants, but they never talk about plants. People have, we as animals, as humans, have an animal-centric view of the world, and we see plants as a beautiful backdrop against which we exist, and we see them as of lesser importance to animals. So we must engender and foster a care and attention for plants in the same way as animals, and maybe we can do that with Rafflesia, as Adrian here has manipulated that slide to show a Philippine eagle of the same equivalent conservation concern as Rafflesia. So we need to tell people that Rafflesia is wonderful and enigmatic and endangered. Next slide. These are some examples of plants that we found at Oxford Botanic Garden have excited people. Rafflesia has the power to excite people. Next. Yeah, exactly. That's a monkey face orchid, Simia. And I wrote an article in Trends in Plant Sciences on this. Next slide. If you haven't already looked, the journal Plants, People, Planet has a series called Flora Obscura. Please do look up Flora Obscura, where some of the plants that we've talked about today feature. And again, and these plants really have the ability to engage people. So some of these plants that we've talked about today in this lecture have been seen and read around the world by millions of people. 8 million people saw one particular one of these. So when we do work on some of these plants, we need to tell the world about it. So when we publish a paper on Rafflesia, we need to excite people, because when we excite people, that's when we can advocate and bring people on board, because not all of the people who have the power to make the decisions that we've talked about are in this room. So when we're trying to lobby local governments, policymakers, decision makers, we need to be able to have the currency of communication to excite them and to influence them and to tell them that the work that we know is important for them as well. And some of the examples from this trip, we just shared some on the screen here, have done that. So people have been fascinated by the plants that are growing here in Sumatra, and we've just put some statistics on the screen to show you the amount of engagement. So on this expedition, we've been sharing some of the highlights from our adventures, and people have been overwhelmed by the flora that you share your home with. Titan arums, Rafflesia, also some of the scary insects that exist in the rainforest. People are very, very excited by these plants. So people care very deeply about these plants if we find the right way to show them. Next slide. And these are some of the heroes in your community in Bengkulu who are pioneers. They're working very hard to conserve Rafflesia and to tell the world about them and about the importance of these plants here. So these people are your ambassadors for Rafflesia. They are ambassadors to the world. They are telling the world about the important plants that you share your home with. Next slide. And so really a big tribute to these people who are the next generation who are going to be the ambassadors to save Rafflesia and to conserve. But I wanted to end, or we wanted to end with this, because we didn't know, this is our first time, for Pat, Adrian and I at least, this is our first time in Bengkulu, and we did not know just how deeply you all care about Rafflesia. So our observations in the Philippines is different, because although we have a huge number of species in the Philippines, people are not so aware of the existence of Rafflesia there. In Indonesia, people care deeply and celebrate these plants, and it seems that every 10 meters you walk in Bengkulu, you see Rafflesia on someone's shirt, outside the hospital, outside the police station, outside the radio station. And so yesterday we had good fun doing a tour of Bengkulu, and everyone looking at us curiously, like, who is this strange man who wants to take pictures of Rafflesia models in our city? But actually, yes, in a way it's funny, but it's also an important point, because when people care about something, they know that it's worth protecting. And so I think there is a very optimistic and hopeful outlook for the conservation of Rafflesia in Bengkulu and elsewhere in Sumatra. And what we now need to do is deploy some of that elsewhere in the world to protect Rafflesia at a global level. And so with that, I would like to thank, on behalf of us all, and I'm representing us as a collective here. We've had an extraordinary expedition for the last 2 weeks. We've been made to feel very welcome. We've met some inspirational people and seen some mesmerizing plants. And so we have cherished memories that we will never forget from our visit. So thank you all very much for having us here to speak to you today. So thank you. And I think now, technology permitting, our very talented Reggie, who's going to put his hand up at the back, he's a bit shy, has produced a wonderful video documentary of our encounters over the last 2 weeks. So we hope you enjoy this. Thank you.
Want to give the narration for this video, no? Giving the narration? No. Okay. Thank you very much, Dr. Chris and team. So this informative and interesting presentation and also documentary video of your journey in Bengkulu. So, ladies and gentlemen, we already heard and also watched the presentation from Dr. Chris and team. I think we will have a vibrant discussion about Rafflesia, parasitic plants, and its biology and also conservation, not only in Bengkulu but also all over the world. So this our opportunity to make me give what suggestion or make be your a question to the expert here from the Oxford and also from the Indonesian. And for this session, I think we will come up with several sessions. I would like to have 3 questions from the participants. So please, you can ask in bahasa. So what's that? We will translate in English. Silakan kalau ada yang ingin bertanya, 1, 2, 3 pertanyaan kepada pembicara yang ada di depan tentang tanaman bunga parasit ya, parasit secara umum, kemudian Rafflesia, dan juga mungkin ya punya pendapat ataupun mungkin hal-hal lain mungkin yang berkaitan dengan apa yang sudah disampaikan sebelumnya. I'm sorry, Dr. Chris, so we just give in dual language, so make the student and also all the audience understand what we want. Oke, silakan adik-adik, mahasiswa ataupun rekan-rekan kolega yang ya, Mr. Jansen, please. Oh, yep. Okay.
Thank you so much for the very nice presentations. I mean, it's so much to digest in like a 1.5 hours. I'm now doing a bit study in the host. Like you said that we got the host specifications. I'm calling my project now this hosting capacity. So the ability of the host to, the tetrastigma to host Rafflesia. So, but what I'm trying to say that I'm intrigued with the, like you said that along the line of the evolutions of the Rafflesia, that you said that at the beginning that the host is not only tetrastigma, the vines family, but also the Euphorbiaceae. So now it is very host specific with the Pitaceae family, with the tetrastigma. Because I'm an ecophysiologist, so I'm trying to see the ecophysiological aspect of the host, Chris. So what I'm doing now is I'm slicing the stem and seeing the anatomy, like you said. I got some pictures, I would like to show it to you. And then also now I'm trying to quantify the water transport, because I'm thinking of probably that will be consequences for the host, because it's hosting huge plants, like you said before that, and because it's water transport or something like that. But then what intrigued me that, like you said, that the diameters of the stems of the roots of the tetrastigma hosting the Rafflesia is quite varied. So it could be a bit large, but it could be very small as well. The one that I saw, the one that you saw in Kamumu, the one next it, I think it's flowering now this week. It's quite a huge flower with a small stem, the host. So I'm trying now to find out whether there is a ecophysiological aspect of the stem. So is there any comments on that, please?
I think it's a fascinating question that you're asking. We also noticed a very small diameter of stem to which the Rafflesia was attached. And 2 points, I think. One is, in the literature, we've talked a lot about Rafflesia either growing in the root or the stem, because, as we know, it's a liane, and so it's not easy to discern. But I suppose one thing I was wondering in reading the literature is if there was a serial anatomy, because at what point in the host does root become stem? Because it's not just abrupt. And so I'd be very interested personally to understand where that junction lies in a liane between the pericicle and the vascular anatomy of a root and a stem. It doesn't just shift. I imagine there's a slight continuum. So I'd be very interested to understand what happens at that junction between root and stem in a vine. And the second point, I think, is it seems to me that endoparasites, the 4 independent lineages, are adapted to living in slow-growing and stable environments, which I think has an adaptive significance for an endoparasite living in a sheltered environment, because some parasitic plants grow on weedy annuals. So some Orobanchi that I work on, they grow on weedy crops, for example. The endoparasites grow in woody, stable environments and are probably quite slow growing. So Rafflesia takes 10 years, we understand, from seed to grow. So it's a slow, steady process, but by and large. So I think it's no coincidence that it grows in a woody, stable environment as well. But I think your studies are fascinating, and I love the idea of understanding more about source-sink relations in analometry.
The stem of the lianas, because the lianas, it is woody stem, but it's quite soft.
Yeah, it's quite pliable.
Quite pliable, and then the amount of water can be transported in one time because it's a huge xylem, huge vessel. So the matter of water transport, it seems it doesn't matter at all for the host. So I'm thinking of something else. So I'm just thinking, oh, this is water enough to study or something like that. So because it's huge vessels, a huge amount of water transported from the soil to the stems, and because the lianas, the tetrastigma also has a huge root pressure to push the water up as well, something like that, please.
Fascinating questions. Yeah. Please, we'd love to.
Yeah, honestly, I'm very much mesmerized on how advanced the research for Rafflesia as well as the host in Bengkulu is. Yeah, but it's a good start. You have it already. But you know, in Rafflesia, what we learned is that when we discover something, we got a lot of questions about it. Yeah, it's really a mysterious plant. Yeah, but you know, my thinking about the water absorption of the Rafflesia from the host, of course, that should be resolved. Which part is the root and which part is the stem? Because all we know, Rafflesia has, tetrastigma has a lot of huge, enormous underground stem. So is it a root or a stem? But probably the reason, because it's inside the vascular tissue or the cell, then it's interconnected. So it doesn't matter where the Rafflesia grow, from the small stem or from the small root or from the big stem, because it depends on the whole plant, not just that stem. Yeah. But yeah, I'm fascinated with how you care deeply with Rafflesia here.
First of all, thank you very much for your question. Really, we have a lot of questions about Rafflesia, right? So I want to share the study of our friend, Dr. Bu Sophie Mercidiwati and Adityo Bichaksono. And they did anatomical study on the tetrastigma vine of Rafflesia, and they found out that anatomically speaking, Rafflesia grows on roots, even the aerial part of the liana. But I think one of the important findings on their study is that Rafflesia originates from the vascular cambium of the tetrastigma. And your study about the water transport on liana will really help us understand how Rafflesia will really grow on tetrastigma. In our experience in Bogor, Bu Sophie demonstrated us how to properly graft Rafflesia, and I think that's very important. Your study will greatly help how to really cultivate Rafflesia ex situ. So thank you very much. Yeah.
This is a beautiful talk, Chris. Thank you. And also Dr. Pat, thank you, Adrian. I would like to add the information about the Rafflesia and the host. And yeah, there is some variation of the host diameter when we take a look the infected tetrastigma. I have the data that the variation is from 2 centimeter to 8 cm. And the average, if we take a look at the data, the average of the root, so-called root infected Rafflesia, is going to be like 3.5 cm. So never higher than the value. This is the one thing. The second is, what about the carrying capacity? So Bergman and also the Pelser from genetic study said there should be a limited population within one host. It's going to be like 15 flower bud. I have data also. There is tend to be population density mortality. If you have a higher population size, then mortality will be higher. So there is modeling indicate that kind of data. So we actually if we take a look within host plant, there should be like limited resources. There is competition over that. For example, Rafflesia arnoldii, the average population size within one host is 7.6 host plant. The bigger size the Rafflesia, the smallest population size. So this is the data that I have will be published, I guess, soon. So the other thing is that we actually lack of study on population ecology. This is very important for developing proper conservation scheme. So I do not know whether we have a collaboration research on long-term ecological monitoring. So between Philippines and Indonesia, for example, and also the Oxford Botanical Garden is going to be good. So before that, I would like to introduce this is a good friend. He is KPPL, one who I guess the founder of KPPL that you mentioned a lot of things. So here is arnoldii. This is the young people from Bengkulu. His background actually in the nurse. Okay. Here is please stand up, arnoldii. So, okay. What I was there. So, yeah. I guess it's my comment and also my introduction to KPPL. So after that, if you have time to discuss, please discuss with him, because he is one of the founder of KPPL. But okay, that's enough for me.
Any other questions from the audience? You can ask in bahasa. Boleh bahasa Indonesia? Adik-adik, bapak, ibu. Oke, ya, silakan.
Thank you for presentation, Mr. Chris. I have question for you. Maybe it's out of topic. After 1 week you in Bengkulu, you have see Rafflesia in Bengkulu, and you have seen several species Rafflesia in Bengkulu. And so, which species do you like the most in Bengkulu? Thank you so much.
I think we should all answer this one. Yeah.
Okay, thank you, Angelica. So please, Mr. Chris, you can answer directly.
I find it really it's a really good question and a really difficult one to answer, because I like them all in different ways. I like the mystery of arnoldii because it's so large. Bengkuluensis is enigmatic and ethereal. But the one that made me most emotional, I think, was Rafflesia camumu, because it was the first one we saw here, and we just arrived, and it had, for me, all of the magic that a Rafflesia encounter can give you. So, and it was unexpected. So, that for me, that was the most magical, and that made me feel the most emotional, I would say.
It's really hard to choose from 3 very beautiful plants, no, Rafflesia. But yeah, they have different uniqueness, no, unique beauty. Bengkuluensis has a very balanced structure, and the color is somewhat lighter than the others. So I like that. And yeah, it has the strongest odor. from the three species that we observe. Of course, yeah, Arnoldi is very much fascinating because of its huge size. And Kemumu, it actually it resembles somehow the Banawana. Wide diaphragm, very much leathery, ramenta, and yeah, all of them are very beautiful. All
of us. Okay, thank you. I think this question is more personally. Yeah, for me, three of species of Rafflesia in Bengkulu, all of them are very interesting for me because this is my first time to see Rafflesia flower in the wild. I just have been seeing the Rafflesia flower only from our garden, from Bogor Botanic Gardens, and only Rafflesia Padma. So as information for all people here that actually I'm working on conservation and systematic of palms, Arecaceae. So for Rafflesia, I'm a newcomer, I think. Newcomer, and how to say, I think it's interesting to continue it because some of our colleagues in Brin also studied on Rafflesia. After this meeting maybe, or after this expedition, I will coordinate my friend who works on Rafflesia to make, how to say, to make more focus on research on Rafflesia. Thank you.
That's a tough question, you know. I've come from the Philippines, and usually our Rafflesia there has a size of 10 to 20 centimeters. The largest on Luzon Island is Rafflesia banawana, which measures 50 centimeters. And it's very common here to see a Rafflesia that measures more than 50 centimeters. But if I will choose from the three, I will choose Rafflesia bengkuluensis. Rafflesia bengkuluensis because when we visited the site, we were accompanied by young people who are volunteers and dedicating their life to conserve, to promote, and create awareness about Rafflesia. And it's a very important endeavor, you know, to fully protect Rafflesia across Southeast Asia. You know, Rafflesia can be a proxy for all plant communities. We do not see plants as significant as animals, but with Rafflesia, people are becoming more aware about our plants, our different species of plants. So with Rafflesia, we can encourage and motivate young people, involve people from different walks of life, from various stakeholders in doing the same action in protecting Rafflesia. So I guess Bengkuluensis is the most very important species for me.
Thank you very much for Dr. Chris and team. So I believe that we still have any more question or topic to be discussed. However, due to limited time, I think this already 12 p.m. So we need to end up our discussion session this afternoon. For the conclusion, there are so much threats for Rafflesia, so such as fragmentation habitat and also stealing from the people. Also, we have learned a lot of information about life cycle of Rafflesia, its morphology, but however, there are still so many mysteries about Rafflesia that we need to learn. So I think never-ending learning about Rafflesia is a good start to do more about that. We need a lot of support from all stakeholders, academicians, researchers, governments, NGO, local people, fund agency as well. So this, I think, collaboration work might be in the next future. University of Bengkulu Forestry Department can collaborate more with Oxford University and also BRIN and also Los Banos, the University of Philippines. So, ladies and gentlemen, we finally come to the end part of this general lecture, the future. Before I close this session, I would like to invite Head of Forestry Department, Vice Dean, and Mr. Agustus Satya to give certificate of appreciation to all our speakers today. So, please, Mr. Edi Suharto, Mr. Jansen, and Mr. Agustus, and all the speakers, please come to in front of this. Okay. Just come here. For the first, please Vice Dean to give, Dr. Chris and Tim, please come here. Okay. Mr. Adrian, you can come there. Okay. Please have a seat again. Thank you, Vice Dean, Head of Department, and Mr. Agustus Satya. Ladies and gentlemen, finally, I would like to thanks to the speaker for the informative and interesting presentation on all the participants of their active participation in this morning until afternoon. Hopefully, this general lecture will be beneficial for everybody. Amin. And please give applause for the speaker and for you all. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Okay. And the last part is a photo session. So I hope all the faculty members and also invited guests to come to the front, and then we take a photo. Ya. Oh, lunchtime. Okay. Ya. Diki. Siapa tadi? Arnoldi, Pak. Just come in front. With these or not? Untuk adik-adik silakan keluar ya, kita sudah selesai. Silakan dibawa apa namanya sampah-sampahnya ya. Oke, makasih ya, makasih.